The overall goal of this procedure is to identify which regions of mRNA are translated into proteins and quantitatively measure protein translation in budding yeast Saccharomyces cerevisiae at the genome-wide level.
Ribosome profiling can be used to monitor the rate of protein synthesis and to study the mechanisms of translational regulation. The main advantage of this technique is that it has much greater sensitivity compared to other methods and allows measuring protein synthesis in vivo at the single codon resolution.
Though this protocol is specific for budding yeast, it is also useful for researchers who are trying to establish ribosome profiling in other model systems, such as mammalian cells and tissues.
Extracts will be prepared from yeast cells grown to mid-log phase. Use a glass holder filter assembly to filter the culture through 0.45 micron membrane filters. Scrape the pellet with a spatula. Flash freeze in liquid nitrogen. And store at -80 degrees Celsius. Put 3 chrome steel 3.2 mm beads into a 1.8 ml stainless steel tube and pre-chill the tube in liquid nitrogen. Add the frozen pellets and cover the steel tube with a silicone rubber cap. Homogenize the sample by cryo-grinding for 10 seconds at 4200 RPM.
To keep the samples frozen during the entire grinding procedure, it is critical to chill the tube in liquid nitrogen for at least 10 seconds in between each grinding.
When the homogenization is done, add 1 ml of lysis buffer that contains cyclohexamide to the sample. Mix well.